So there are several challenges I can think of detecting pathogens from food. To give you an example, like the first challenge would be separating pathogens and toxins from food matrices. Food matrices are very complex, and sometimes the microorganisms remain attached to the food matrices. It's very difficult to remove them, and therefore, if you develop detection platforms that may not be able to detect that matrix. The second challenge would be the pathogens we find in the food. Sometimes they are injured or stressed, and so as a result, they may not grow properly, and so our detection platform or sensors may not be able to find them. So that's often a challenge when you find pathogens coming from a processed product, for example, or being stored at cold storage. And so those pathogens present a different challenge to the microbiologist for detection of those organisms. And the third common problem could be the concentration of bacterial cells or toxins in food. So if the food is processed, most of the time we don't expect a high level of those pathogens or the bacteria in the food. So therefore, the pathogens need to grow or build their numbers before the sensors or detection platform can detect them. And so those are the three major challenges I see we face routinely for detection of those organisms from food. So I can talk about three major sensors we are developing. One of them called light scattering sensor. Essentially, in this case, we use a laser to point towards bacterial colony which grows on the petri dish. And as a result, when the laser hits, it forms a scatter signature or fingerprint. And so based on that fingerprint, we could identify a new or unknown organism if they are present in that food. So for example, Listeria or Salmonella, they would produce a unique fingerprint, and once we match that fingerprint image with the database, we would be able to readily identify that organism. Two other methods we are developing. One is called fiber optic sensor. In this sensor, we are using antigen antibody sandwich format on an optical waveguide. And as bacteria binds or toxin binds to this specific antibody molecule or receptor molecule immobilized on the fiber, and that would yield a specific fluorescent signal. And so with the help of detector, we would be able to get that signal comes as a fluorescence molecules become excited. So this cannot differentiate live or dead cells, but it can detect specific pathogen based on the antibody or receptor molecules we have on the fiber. And the third one is called cell-based sensor, and this sensor essentially based on the mammalian cells or live cells, since pathogens and toxins interact with those live cells. And so our goal is that when these toxins or pathogens bind to these mammalian cells, and the cells would respond in a form, either they are dead or they will be lysed or they will be emitting some kind of signal which the detector can detect. So these are the three major sensors we are developing. All have some advantages and disadvantages, and the application would be also broad. Some can provide results within minutes to hours. Some may take several hours to days. So depending on the sensors, light scattering sensor, it takes about a day or day and a half. Fiber optic sensors results can be obtained within 4 to 6 hours, and cell-based sensors, results can be received within an hour or so. So all sensors has a different application for different pathogens and toxins. So our immediate goal is to see if these sensor platforms can be applied for foodborne pathogen or toxin detection and see commercial availability or success. So one of the future goal would be we need to verify, make sure these sensors work with real world food samples or real samples. So that would be immediate goal, and obviously the next goal would be if these platforms or sensors can detect these pathogens very quickly or rapidly, so that we can ensure food safety and so the consumers can be protected from foodborne diseases. And obviously, if the commercial success is there, that's going to benefit all of us.